Hello dear students, welcome to this program. Today we are discussing an interesting topic which is about chromatin structure, euchromatin, heterochromatin, constitutive and facultative heterochromatin. Before discussing them in detail, let us first know its objectives. The main objectives are to understand what is chromatin and its structure, brief description about euchromatin and heterochromatin, constitutive and facultative heterochromatin. Chromatin. The eukaryotic cell cycle produces remarkable changes in the structure of chromosomes. In non-dividing eukaryotic cells in G0 and those in interphase, that is G1S and G2, the chromosomal material chromatin is amorphous and appears to be randomly dispersed in certain parts of the nucleus. Chromosomes become much more condensed during prophase of mitosis, taking the form of a species-specific number of well-defined pairs of sister chromatids. Chromatin consists of fibers containing protein and DNA in approximately equal masses, along with a small amount of RNA. The DNA in the chromatin is very tightly associated with proteins called histones, which package and order the DNA into structural units called nucleosomes. Histones are small basic proteins found in the chromatin of all eukaryotic cells. Histones have molecular weights between 11,000 and 21,000 and are very rich in the basic amino acids, that is arginine and lysine. Together, these make up about 1/4 of the amino acid residues. The amino acid sequences of histones, particularly H3 and H4, have undergone very little change over long periods of evolutionary time. Why are histones so highly conserved? One reason is histones interact with the backbone of the DNA molecule, which is identical in all organisms. In addition, nearly all the amino acids in a histone molecule are engaged in an interaction with another molecule, either DNA or another histone. Also found in chromatin are many non-histone proteins, some of which help maintain chromosome structure, others that regulate the expression of specific genes. About 10% of the chromatin in a typical eukaryotic cell is in a more condensed form than the rest of the chromatin. This form, heterochromatin, is transcriptionally inactive. Heterochromatin is generally associated with particular chromosome structures, that is, the centromere. For example, the remaining less condensed chromatin is called euchromatin. Now coming to the structure of chromatin. In the early 1970s, it was found that when chromatin was treated with non-specific nucleases, most of the DNA was converted to fragments of approximately 200 base pairs in length. In contrast, a similar treatment of naked DNA, that is, DNA devoid of proteins, produced a randomly sized population of fragments. This finding suggests that chromosomal DNA was protected from enzymatic attack except at certain periodic sites along its length. It was presumed that the proteins associated with the DNA were providing the protection. In 1974, using the data from nucleus digestion and other types of information, Roger Kornberg, then at Harvard University, proposed an entirely new structure for chromatin. Kornberg proposed that DNA and histones are organized into repeating subunits called nucleosomes. Each nucleosome contains a nucleosome core particle consisting of 146 base pair of supercoiled DNA wrapped almost twice around a disc-shaped complex of 8 histone molecules. The histone core of each nucleosome consists of two copies each of histones H2A, H2B, H3 and H4 assembled into an octamer. The remaining histone, that is type H1, resides outside the nucleosome core particle. The H1 histone is referred to as a linker histone because it binds to the part of the linker DNA that connects one nucleosome core particle to the next. Together, the H1 protein and the histone octamer interact with about 168 base pairs of DNA. H1 histone molecules can be selectively removed from the chromatin fibers by subjecting the preparation to solutions of low ionic strength. When H1 depleted chromatin is observed under the electron microscope, the nucleosome core particles and naked linker DNA can be seen as separate elements which together appear like beads on a string. Our understanding of DNA packaging has been greatly advanced in recent years by dramatic portraits of the nucleosome core particle obtained by X-ray crystallography. The 8 histone molecules that comprise a nucleosome core particles are organized into 4 heterodimers, 2 H2A, H2B dimer and 2 H3H4 dimers. Dimerization of histone molecules is mediated by the C-terminal domain, which consists largely of helices folded into a compact mass in the core of the nucleosome. In contrast, the N-terminal segment of each core histone and also the C-terminal segment of H2A takes the form of a long flexible tail that extends past the DNA helix and into the surroundings. These tails are targets of a variety of covalent modifications. In addition to the 4 conventional core histones discussed above, several alternate versions of the H2A and H3 histones are also synthesized in most cells. DNA and core histones are held together by several types of non-covalent bonds, including ionic bonds between negatively charged phosphate of the DNA backbone and positively charged residue of the histones. The two molecules make contact at sites where the minor groove of the DNA face inward towards the histone core, which occur at approximately 10 base pair intervals. In between these points of contact, the two molecules are seen to be separated by considerable space, which might provide access to the DNA for transcription factors and other DNA binding proteins. For many years, histones were thought of as inert structural molecules, but as we will see, these small proteins play critically important roles in determining the activity of the DNA with which they are associated. It has also become evident that chromatin is a dynamic cellular component in which histones, regulatory proteins, and a myriad variety of enzymes move in and out of the nucleoprotein complex to facilitate the complex task of DNA transcription, compaction, replication, recombination, and repair. The assembly of nucleosomes is the first important step in the compaction process. With a nucleotide spacing of 0.34 nanometer, the 200 base pair of a single 10 nanometer nucleosome will stretch nearly 70 nanometer if fully extended. Consequently, it is said that the packing ratio of the DNA of nucleosomes is approximately 7:1. Now moving to higher levels of chromatin structure. A DNA molecule wrapped around nucleosome core particles of 10 nanometer diameter is the lowest level of chromatin organization. Chromatin does not, however, exist within the cell in this relatively extended beads on a string state. When chromatin is released from nuclei and prepared as physiological ionic strength, a fiber of approximately 30 nanometer thickness is observed. Despite more than 2 decades of investigation, the structure of the 30 nanometer fiber remains a subject of debate. Two models in which the nucleosomal filament is coiled into the higher order, thicker fiber, the models differ in the relative positioning of nucleosomes within the fiber. Recent research favors the zigzag model in which successive nucleosomes along the DNA are arranged in different stacks and alternating nucleosomes become interacting neighbors. Regardless of how it is accomplished, the assembly of the 30 nanometer fiber increases the DNA packing ratio an additional 6-fold or about 40-fold altogether. Maintenance of the 30 nanometer fiber depends on the interaction between histone molecules of neighboring nucleosomes. Linker histones and core histones have both been implicated in higher order packaging of chromatin. Core histones of adjacent nucleosomes may interact with one another by means of their long, flexible tails. Structural studies indicate, for example, that the N-terminal tail of an H4 histone from one nucleosome core particle can reach out and make extensive contact with both the linker DNA between nucleosome particles and the H2A, H2B dimer of adjacent particles. These types of interactions are thought to mediate the folding of the nucleosomal filament into a thicker fiber. In fact, chromatin fibers prepared with H4 histones that lack their tails are unable to fold into higher order fibers. The next stage in the hierarchy of DNA packaging is thought to occur as the 30 nanometer chromatin fiber is gathered into a series of large supercoiled loops or domains that may be compact into even thicker, that is 80 to 100 nanometer fibers. The DNA loops are apparently tethered at the bases to proteins that are part of an organized nuclear scaffold or matrix. Normally, loops of chromatin fibers are spread out within the nucleus and cannot be visualized, but their presence can be revealed under certain circumstances. The mitotic chromosome represents the ultimate in chromatin compaction. 1 micrometer of mitotic chromosome length typically contains approximately 1 centimeter of DNA, which represent a packing ratio of 10,000:1. This compaction occurs by a poorly understood process. Now coming to heterochromatin and euchromatin. After mitosis has been completed, most of the chromatin in highly compacted mitotic chromosome returns to its diffuse interface condition. Approximately 10% of the chromatin, however, generally remain in a condensed, compacted form throughout interphase. This compacted, densely stained chromatin is seen at the periphery of the nucleus. Chromatin that remains compacted during interphase is called heterochromatin to distinguish it from euchromatin, which returns to a dispersed state. When a radioactively labeled RNA such as 3H uridine is given to cells that are subsequently fixed, sectioned and autoradiographed, the clumps of heterochromatin remains largely unlabeled, indicating that they have relatively little transcriptional activity. The state of a particular region of the genome, whether it is euchromatin or heterochromatin, is stably inherited from one generation to the next. Heterochromatin is divided into two classes. That is, constitutive heterochromatin, that means it remains in the compacted state in all cells at all times and thus represents DNA that is permanently silenced. In mammalian cells, the bulk of the constitutive heterochromatin is found in the region that flanks the telomeres and centromere of each chromosome and in a few other sites such as the distal arm of the Y chromosome in male mammals. The DNA of constitutive heterochromatin consists primarily of repeated sequences and contains relatively few genes. In fact, when genes that are normally active move into a position adjacent to heterochromatin, that is having changed position as the result of transposition or translocation, they tend to become transcriptionally silenced, a phenomenon known as position effect. It is thought that heterochromatin contains components whose influence can spread outward a certain distance, affecting nearby genes. The spread of heterochromatin along the chromosome is apparently blocked by specialized barrier sequences, that is, boundary elements in the genome. Constitutive heterochromatin also serves to inhibit genetic recombination between homologous repetitive sequences. This type of recombination can lead to DNA duplications and deletions. Now coming to facultative heterochromatin. It is a chromatin that has been specifically inactivated during certain phases of an organism's life or in certain types of differentiated cells. An example of facultative heterochromatin can be seen by comparing cells of a female mammal to those of a male. The cells of males have a tiny Y chromosome and a much larger X chromosome. Because the X and Y chromosomes have only few genes in common, males have a single copy of more genes that are carried on the sex chromosome. Although cells of females contain two X chromosomes, only one of them is transcriptionally active. The other X chromosome remains condensed as a heterochromatic clump called a bar body after the researcher who discovered it in 1949. Formation of a bar body ensures that the cells of both males and females have the same number of active X chromosomes and thus synthesize equivalent amounts of the products encoded by X-linked genes. X chromosome inactivation. Based on her studies of the inheritance of coat color in mice, the British geneticist Mary Lyon proposed the following in 1961. Heterochromatinization of the X chromosome in female mammals occurs during early embryonic development and leads to inactivation of the genes on that chromosome. And heterochromatinization in the embryo is a random process in the sense that the paternally derived X chromosome and the maternally derived X chromosome stand an equal chance of becoming inactivated in any given cell. Now coming to the histone code. Cells contain a remarkable array of enzymes that are able to add chemical groups to or remove them from specific amino acid residues in the histone tails. Those residues that are subject to modification, most notably by methylation, acetylation or phosphorylation. The past few years has seen the emergence of a hypothesis known as the histone code, which postulates that the state and activity of a particular region of chromatin depend on the specific modifications or combinations of modifications to the histone tails in that region. Comparison of the nucleosomes present within heterochromatin versus euchromatin domains revealed a striking difference. The lysine residue at the position 9 of the H3 histone in heterochromatin domains is largely methylated, whereas this same residue in eukaryotic domains tends to be unmethylated, although it's often acetylated. Removal of the acetyl groups from H3 and H4 histones is among the initial steps in conversion of euchromatin into heterochromatin. The correlation between transcriptional repression and histone deacetylation can be seen by comparing the inactive heterochromatin X chromosome of female cells which contain deacetylated histones to the active euchromatin X chromosome whose histones exhibit a normal level of acetylation. Histone deacetylation is accompanied by methylation of H3K9, which is catalyzed by an enzyme, that is a histone methyltransferase, that appears to be dedicated to this and only this particular function. This enzyme called SUV39H1 in humans can be found localized within heterochromatin, where it may stabilize the heterochromatin nature of the region through its methylation activity. The formation of a methylated lysine at the position 9 endows the histone H3 tail with an important property. It becomes capable of binding with high affinity to proteins that contain a particular domain called a chromodomain. The human genome contains at least 30 proteins with chromodomains, the best studied of which is heterochromatin protein 1, that is HP1. Acetylation of lysines on both histones H3 and H4 is closely correlated with transcriptional activation. The effects of methylation of H3 and H4 lysines depend strongly on which of these residues is modified. For example, methylation of lysine 9 of histone H3 is typically present in heterochromatin and associated with transcriptional repression. Methylation of H3K27 and H4K20 is also strongly associated with transcriptional repression, whereas methylation of H3K4 and H3K36 is associated with activation. Just as there are enzymes that add each of these groups, there are also enzymes, that is deacetylases, demethylases and phosphatases, that specifically remove them. It's all about this topic. Hope you understood.